Hi, thanks for coming, everyone, and thank you for the introduction stuff. So my name is Joy. I am a graduate student in the Hakohen Lab at the Broad Institute. And tonight I'm going to talk about how immunotherapy is awakening the force within and bringing a new hope to cancer treatment. So first, I'd like to put everything in context. So imagine that the circle represents all the different types of cancer treatment. This includes things like therapy, radiation therapy, and chemotherapy. Immunotherapy, which is a little bit newer on the scene, is a subset of all these different types of cancer therapies. And specifically in this talk, I'm going to be talking about immune checkpoint inhibitors. And this is a specific type of immunotherapy. So for the duration of this talk, when I say immunotherapy, what I really mean is this immune checkpoint inhibitor. And these three words might seem kind of random or nonsensical right now, but we will go through it during the talk. And you will leave with an understanding of what the immune checkpoint inhibitor does and also what current researchers are looking at. So in order to understand cancer immunotherapy, we need to understand both cancer biology and immunology, and cancer immunotherapy lies within the intersection of these two. So this is the roadmap for the evening. First, we're going to briefly talk about what is so great about this cancer immunotherapy, and then how cancer cells are different from our normal cells, how immune cells recognize abnormal cells, and then we will take a brief intermission. When we come back, we'll talk about how these immune checkpoint inhibitors actually work, and last, what researchers are currently working on in this field. So first, what is so great about cancer immunotherapy? Near the end of last year, you probably saw some of these headlines in the news about how former President Jimmy Carter was diagnosed with advanced melanoma that has spread to both his liver and his brain. However, a few months later on immunotherapy, he has been declared cancer-free. And I especially like the title of the CNN article that asks if this is miracle or just science. So what is so great about immunotherapy? The first immune checkpoint inhibitor called ipilimumab, or simply we just say IPI, was approved by the FDA in 2011. And while IPI worked really well for some people, the fraction of people who actually respond to this treatment was pretty low, and it also gave people very bad side effects. The newer generation of immunotherapy came out on the market in 2014, so this is a very recent development. And there were two different new checkpoint inhibitors, pembrolizumab and nivolumab, and these were both approved for the treatment of melanoma. And currently, there's more than 150 different clinical trials for using immune checkpoint inhibitors to treat different cancers such as lung cancer, skin cancer, kidney, ovarian, and bladder cancers. So it's a very exciting field. So immunotherapy was initially approved for melanoma. So I'm going to talk a little bit about what it has done for melanoma treatment. Advanced melanoma is extremely difficult to treat, and the one-year survival rate is between 35 to 54%. This means that someone who was diagnosed with advanced melanoma, 1 year later, the percentage of people who have been diagnosed is only 35 to 54%. And on treatment with a single immunotherapy, this percentage jumps to 74%. And there's some reports of combining different immunotherapies where the 1 year survival then jumps to 94%. So this is a huge improvement. And these studies are very new, and the results are coming in right now. So we actually don't have good 5-year survival rates, which is what people like to look at, because this drug was only approved in 2014. And the really great thing about the 2014 drugs is that the side effects are actually very mild. They tend to include things like fatigue, itching, and rash. And the more serious side effects include kidney failure and colitis, but these are fairly rare. So for this talk, I'm going to focus on the new drugs that were introduced in 2014 instead of the 2011 ones. Okay, so let's start with a little bit of cancer biology. How are cancer cells different from normal cells? Well, the biggest difference is this uncontrolled cell division, but what causes it? So let's look at what a cell from your body looks like. So we know that every cell contains DNA. And the DNA isn't just free floating in the cell, it's contained in this special package in the cell called the nucleus, and that's shown as this red sphere in this picture. So in the nucleus, DNA is organized into 23 pairs of chromosomes. And when we zoom in further, we can see the double helical structure of DNA. So this is a cartoon representation of DNA, and in case you're curious, here is a nice animation of what the molecular structure of DNA looks like. We can think of DNA structure as a ladder that you've then twisted into this double helix. And DNA is important because it contains the information that's required to make all the proteins in your body. And proteins are really the workhorses of the body, and they facilitate all the different chemical reactions that's necessary for life. So the information that's encoded in DNA are actually found on the rungs of this ladder. There's 4 different molecules that make up these rungs, and to simplify things, scientists have given them one-letter designations, and these letters are A, C, G, and T. So in DNA, the letter C always binds to the letter G, and the letter T always pairs with the letter A. So if you look at one strand of DNA, if you read what those letters are, you automatically know what the other strand is going to say. And we also see that there's 2 smaller molecules, the C and T, and 2 bigger molecules, the A and G. So the smaller molecule always pairs with the larger molecule, and this gives DNA its constant width. Sections of DNA that encode for proteins are called genes, and these genes are made into RNA by a protein complex called the RNA polymerase. So this is what an RNA polymerase actually looks like. The green here shown is the DNA, and the yellow is the RNA that the RNA polymerase is currently making. And everything else that's kind of messy looking in the picture is the actual RNA polymerase. But here I drew it as this kind of little face that's eating DNA because it's a little easier to see. And this is what the structure of RNA looks like. We can see that it's actually very similar to DNA, but unlike DNA, there's only one strand. After all this RNA is made, another specialized machinery in the cell called the ribosome comes and it reads the RNA and it creates new protein from the RNA strands. The specialized machinery called the ribosome is actually made up of both RNA and protein. And this process of going from DNA to RNA to protein is called the central dogma of biology. And if you attended Vinny's talk a few weeks earlier, you might remember that she also talked about the central dogma. And also here is what the ribosome looks like. It's also a very complicated structure, and it's made up of this large subunit and a small subunit. Okay, so occasionally DNA becomes damaged, and we call these damage mutations. So we can think of mutations as a typo in DNA. So originally, maybe the DNA said A at a position, and it has been converted into G. What causes these typos? A very common source is UV light. This could be just from the sunlight or tanning beds. Ionizing radiation, such as X-rays or gamma rays, can also damage your DNA and cause mutations. We also have mutagens in the environment, and this includes cigarette smoke as a common source, and also some of the urban pollutants that Shanina talked about in her talk a few weeks ago. DNA damage or mutations could actually occur just during regular cell replication when the protein that creates new DNA strands make a mistake. So when there's a mistake in DNA, this mistake is propagated to RNA by RNA polymerase, and when the ribosome comes and reads the RNA, it can sometimes create a protein that is now broken and non-functional. Why are mutations bad, especially in the context of cancer? Well, these mutations can occur in genes that encode for tumor suppressors. And tumor suppressors tell cells to stop dividing when DNA damage is detected. And also, if the DNA damage is too severe, these tumor suppressors tell the cell to die instead of making more copies of these broken cells. So here I'm going to talk about p53, which is a very well-known tumor suppressor, and is also the most frequently mutated gene found in cancer. So how does p53 work? Well, before a cell decides that it's going to make a copy of itself, it goes through several checkpoints to look at whether or not it should be dividing. So during one of these checkpoints, p53, which is this little guy in a hat here, asks all of its minions, which are these little yellow blobs, whether or not it's clear to divide. these little yellow blobs, which are different proteins, check for different types of DNA damage, and they all report back to p53 and say, okay, it's clear to divide, after which p53 will then allow the cell to proceed with division. However, once in a while, one of these minions might actually detect something, for example, UV damage. So when p53 realizes that there's damage, it tells the cell to stop dividing immediately and to fix and to activate all of these new proteins that will fix the DNA damage before the cell can replicate again. So of course, I drew p53 as this guy in a hat here because it's easier to see, but the actual structure of p53 is a bit more complicated. It actually binds to DNA when it's activated as 4 different copies of itself. And this is how it can turn on the production of new proteins that will fix DNA. When a mutation occurs in p53, it can inactivate this protein. So now when one of its minions comes to it and says, UV damage is detected, the cell should stop dividing, p53 can't relay that message to the cell anymore. And this leads to the accumulation of more and more mutations in the cell. So here's a fun aside for you about p53. So if we assume that every single cell has the same probability of becoming cancerous, then if someone has more cells, then that should mean that you're more likely to get cancer, right? So people have found out that this is actually true within humans. So the more cells that you have, the bigger you are, actually the more likely you are to die from cancer. But this relationship breaks down between different species. So for example, elephants are much bigger than we are. They have much more cells than we do. But the elephant mortality from cancer is actually only around 5%, and human mortality from cancer is around 11 to 25%. So this paradox was actually resolved in 2015, just last year, when researchers sequenced the elephant DNA and found out that they have actually 20 different copies of p53, and we only have 1. So this means that an elephant can have mutations that knock out 19 copies of their p53, and they will still have 1 left. And this can explain why they are so much more resistant to cancer than we are. Okay, so back to these mutations. Why is it bad when mutations accumulate? Well, mutations may convert genes called proto-oncogenes into oncogenes. And oncogenes are what turns a cell into a cancer cell. So proto-oncogenes are genes in the body that are frequently involved in things like cell division. And when a mutation comes along, it can convert a proto-oncogene into what you can think of as this evil twin. So now the proto-oncogene becomes this broken protein that turns the normal cell into this rapidly dividing tumor cell. So I'm going to introduce a very important proto-oncogene to you called BRAF. And BRAF is a very important protein involved in cell division, and BRAF is mutated in around 50% of melanomas. So BRAF sits in what is known as a signaling pathway. And the signaling pathway consists of many different proteins, and you can think of these proteins as kind of playing telephone with each other. So a signal comes upstream, and each protein tells the message to the next protein in the pathway. So here are 4 important members of this particular signaling pathway. Their specific names are not that important, and I've given BRAF here a mustache and a little monocle just to make him a little bit easier to remember. So some signal comes upstream that says the cell should divide. And eventually this signal is relayed to RAS, and RAS tells BRAF the cell needs to divide. BRAF then tells the next member the cell needs to divide. And then ERK then tells the next member, MAPK, that the cell should divide. And eventually the signal is relayed all the way down the chain to a lot of different proteins, and this eventually results in cell division. So when BRAF becomes mutated, it actually gets stuck in this on position. So here's the mutated BRAF. And now, instead of actually relaying the message from RAS, it's just constantly telling the next member in the pathway to divide. So here we see that RAS is asleep. It's not getting a signal about cell division at all. But the broken BRAF, which is stuck in the on division, is still telling ERK that the cell wants to divide. And ERK doesn't know any better. All it knows is that it's receiving the signal. So the signal still propagates down the chain, resulting in uncontrolled cell division. And here's what BRAF actually looks like. So why is uncontrolled cell division bad? maybe we actually want more of some of these cells. So I used to think actually, maybe if I had more brain cells, that would make me smarter. So maybe we actually want more of these cells. Well, the problem is that cancer cells are actually no longer functional cells. Instead of doing their jobs, all they do is suck up nutrients and crowd out the healthy cells, and this can cause the healthy cells to die. And also, in later stages, cancer cells can travel to vital organs and disrupt the function of these vital organs, leading to death. So here's another example with melanoma. So we know that melanoma starts on the skin. However, when melanoma gets more advanced, these cancer cells can actually spread through the bloodstream to different parts of the body. And a very common organ where melanoma spreads to is the liver. So here in this picture, the melanoma cells in the liver have been stained red. So all the red cells that you see in this, all the red spots are cells that are actually from melanoma, and the rest of the cells are your functional liver cells. So we know that cancer occurs randomly throughout our body, and we want to get rid of these cancer cells. And how do we actually do that? Well, it turns out that the immune system actually comes to our rescue and most of the time manage to kill most abnormal cells before they have a chance to become cancerous. So to summarize this section, we saw the flow of information from DNA to RNA to proteins. And we saw that a mutation in DNA can be propagated to RNA and to proteins, and this can result in a non-functional protein. We saw that p53 functions as a very important tumor suppressor. However, frequently in cancer, mutations knock out p53, so p53 is no longer functional. And this can let mutations accumulate. When mutations accumulate in proto-oncogenes, this can convert them into oncogenes, and this can lead to uncontrolled cell division and cancer. So next up, we're going to talk about how the immune system works and how it can help us get rid of these abnormal cells. So any questions before we move on? All right. So next, we are going to introduce you to the immune system. And before we do that, we need to enter a bit of a new mindset, which is that while we all like individuality and creativity, there is no place for that in the human body. Every cell in your body has a very specific function, and it cannot be allowed to deviate from this function. So here's a little immune cell, and because its job is to keep peace and order throughout the empire, I've given it a little stormtrooper hat. So the primary function of the immune system is to determine what does and does not belong in the body. And it does this by recognizing different antigens. An antigen is defined as any molecule that is capable of inducing a response from the immune system. And antigens are frequently derived from pieces of bacteria or viruses that are found in our body. And they could consist of different proteins or even sugars and fats. And the immune system has these specific receptors that fit into the same shape as these antigens. And when the immune receptor fits with the antigen, this elicits an immune response. So for this talk, we will focus on a specific type of immune cell called the T cell. T cells are really important for finding and killing cells that have been infected with viruses and also cancer cells. And each T cell has a unique receptor that are shown as kind of its little arms on the side of its body. And each receptor recognizes a specific antigen. So these receptors, you can almost think of them as a unique fingerprint on the T cell because each T cell has a different one in the body. And this is what a T cell actually looks like. But for this talk, it's a little easier to think of them just as mini stormtroopers. So how are T cells actually made, and how do we get all these different receptors that's capable of recognizing so many different shapes? T cells began their long journey to maturation very early in our life. In the 8th or 9th week of gestation, these progenitor T cells, which are cells that will eventually become T cells one day, migrate to the thymus. And the thymus is found roughly in this region between our lungs. It's not an organ that we usually talk about. So in the thymus, the progenitor T cells will create their own unique T cell receptors. In order to do this, the T cells actually shuffle their different DNA segments. And T cells are one of the really unique cell types in your body because it actually changes its own DNA. All the other cells in your body have the exact same copies of DNA. So the T cell creates the T cell receptor from two different chains called the alpha chain and the beta chain. These are two separate proteins that each make up two halves of the T cell receptor. The alpha chain is made from three different segments called the C, J, and V segment. And the beta chain is made from four different segments called the C, J, D, and V. So our DNA actually contains 54 different V segments, 61 different J segments for the alpha chain. So if you think of all the possible alpha chains there are, when the T cell randomly picks a V and randomly picks a J, we get 54 times 61. And there's many different copies of C, J, D, and V for the beta chain as well. So we can multiply all of these different numbers together. But the actual number of possible combinations is even bigger than that, because we add in certain things called the joining flexibility, and also the T cell randomly inserts some stuff into these sequences to create more randomness. And when you do all the math, you end up with more than 10 trillion possible sequences. And this is a 1 followed by 13 different zeros. However, most of these sequences are actually non-functional. So imagine if you decided to write a novel, and you do this by closing your eyes and pounding your keyboard with your fists. And you do this 10 trillion times. How many great novels have you written? Well, this is similar to what the T cell is currently doing. So we started with this huge pool of immature T cells with these random receptors of different shapes. And now we need to get rid of the broken ones. So which ones are actually broken? Well, the ones that are broken are the ones that don't work at all. And also the ones that recognize normal antigens found in the body. And these antigens are called self-antigens. So we have our own proteins that are normally found in the body, and sometimes these proteins break up and they have different shapes that a random T cell receptor might actually recognize. So we want to get rid of these T cells, because if they recognize a self-antigen, then they could attack our own cells, and this could lead to something like an autoimmune disease. So we have specialized cells in the thymus that actually display a lot of these self-antigens on the cell surface. And it checks each T cell to make sure that it doesn't recognize a self-antigen. If a T cell ends up recognizing a self-antigen, this special purple cell says, well, unfortunately, you recognized a self-antigen, and now you must die because you cannot enter the rest of the body. So we've eliminated all these broken T cell receptors. And now our one remaining T cell gets its new stormtrooper helmet, and it's allowed to get into the blood, and it can circulate through the bloodstream and monitor the cells in our bodies. This process that creates these unique T cell receptors eventually generates around 25 million different T cells in our body. So now that we have these T cells, how do the cells of the body actually identify themselves to the immune system and convince the T cells that they're functioning properly? So when a normal cell in the body creates a protein, sometimes it chops these proteins up into different fragments, and then it loads this fragment onto a complex of proteins called the major histocompatibility complex 1, or simply the MHC1. So MHC1 binds these protein fragments, and then the cell displays them on the cell surface. So here we see them as these little hands that's holding the purple circles. And the purple circles indicate that it's a self-antigen. So when the T cell comes by asking for identification from these cells, and the cells are displaying self-antigens, the T cell will not recognize any of these self-antigens, because we saw that in the previous step, we had eliminated any T cell that recognized self-antigens. So the T cell, when it meets a normal cell, will leave the normal cell alone. However, when your cell gets infected by a virus, the virus takes over the cell's production capacity, and instead of making normal proteins inside the cell, now this infected cell is just pumping out more and more viral proteins. So it's still chopping up these viral proteins into little fragments and loading them onto MHC1. Except now, instead of the self-antigens that it used to be loading, it's holding these viral protein fragments onto MHC1. And when the T cell comes by, it's able to recognize the viral protein fragments because it's a foreign antigen. And here's what this complex actually looks like. So the blue proteins are the T cell receptors, and the orange proteins are the MHC1 complex from the infected cell. The little bit of red that you see in that picture is the antigen that is being bound to the MHC1. So when the T cell recognizes a cell that's presenting this foreign antigen, it will then shoot out specialized proteins that will kill the cell. And these proteins punch holes in the cell membrane. And the reason that we want to kill cells that have been infected with viruses is because this stops the cell from making more and more viruses, and it limits the spread of infection. However, sometimes these activated T cells go a little too far, and it causes a lot of collateral damage to the surrounding normal tissue. To limit this collateral damage, normal cells can deactivate T cells using this protein called the programmed death ligand 1, or PDL1. And PDL1 binds to a receptor on the T cell that's called the PD1. So these are the little blue ball and receptor. And when this binding occurs, the T cell turns off. So PD1 is known as an immune checkpoint because here the immune system is checking whether or not it should be on. And PD1, PDL1 binding plays a very important role in regulating the immune system and preventing it from going too far. So in this previous section, we saw the process which our body generates up to 25 million different T cell receptors, and how our body eliminates T cells that can recognize self-antigens in this process. We saw how T cells can recognize cells that have been infected by viruses and kill them, and how normal tissue can prevent collateral damage by using the PD1 and PDL1 system to turn the T cells off. So next up, we will talk about how the immune system actually recognizes cancer cells and how immune checkpoint inhibitors actually work. So now we are going to talk about how these immune checkpoint inhibitors actually work, now that we've understood a bit about cancer biology and immunology. So like we mentioned before, cancer cells tend to have many different mutations, and these result in mutated proteins that are all telling the cell to keep dividing. And this is especially true in cancers such as skin and lung cancer, because these are the result of a lot of mutagenesis resulting from either UV light or cigarette smoke. So because the cancer cell has all these different mutated proteins, when they chop them up and put fragments of it onto MHC1, they are no longer, they don't look like the self-antigens anymore. Because of these mutations, now they are what we call neoantigens, and these can be actually recognized by the immune system. So here, our stormtrooper T cell comes by, and it binds onto the neoantigen found on the cancer cell, and it recognizes it as a rebel scum. And once this recognition occurs, the T cell can then kill the cancer cell using the same method that it uses to kill a virally infected cell. However, cancer cells can also use the PDL1 and PD1 pathway to escape immune killing. So here was the normal use of PD1 and PDL1, where a normal cell can tell the T cell to turn off to prevent collateral damage. However, a cancer cell can also use a lot of copies of PDL1 on the cell surface to play this Jedi mind trick on the T cell. So even though the T cell has clearly recognized the neoantigen, the cancer cell can use PDL1 to tell it, these are not the antigens you're looking for. And when this happens, the cancer cell is able to escape immune killing. So what immune checkpoint inhibitors actually do is that they bind to either PD1 or PDL1, and this prevents the cancer cell from using this pathway to turn off the T cell. So now that the Jedi mind trick no longer works, the T cell is again able to kill off these cancer cells. So what are researchers currently working on in the field of immune checkpoint inhibitors? So I'll tell you a little bit about my research at the Broad Institute. And currently, we're trying to figure out why some patients who initially had a great response to immunotherapy relapsed just several months or even years later. So here's one patient. This patient went on immunotherapy over the course of 12 months, and we see that during the first 6 months, this person had a great response to immunotherapy. but then this patient relapsed and her cancer stopped responding at month 6. So at each of these time points, we took a biopsy from this patient from one of the lesions from the melanoma. And we also took a sample of normal blood. So from these 2 samples, we can extract the DNA sequences. And the reason why we want DNA from both the normal blood and also the biopsy is so that we can compare the 2 and see where the 2 differ, so we can identify the mutations that occurred in the cancer. So for this patient, we have many different biopsies throughout this time course. And the question we want to ask is what happened in the tumor between these time points to cause a relapse? So now that we have all this DNA, we employed a technique called shotgun sequencing, and we used a bunch of different computer programs to figure out what actually happened in the DNA. So here's a brief explanation of what shotgun sequencing actually does. So imagine that this sentence, which contains information, is our DNA. And our DNA is very, very long. So normal sequencing methods can't actually read this very, very long sentence. So what we do is we make many different copies of the sentence and we chop it up into small pieces. And then we can read the small pieces of sentences and use a computer to align these small pieces back to the original sequence. So now if there's a mutation in the DNA, or in this case a typo in our sentence, when we chop them up, there's still enough information left for the computer to align it back to the original sequence, and we can say, this I has been turned into an A. So we can then go through the DNA and find out where in this DNA sequence all these mutations occurred. So there's over 3 billion different bases or letters in the human genome. So we definitely can't do this by eye. So we have another computer program to do this for us. And here are some actual DNA sequences from my research where we discovered a mutation at this particular position in the DNA. And you might also notice a few little blips in the sequences here where they don't match up with the normal sequence that's in color. And these are considered to be sequencing errors. So when we use a sequencing machine to read the DNA, it doesn't do this perfectly, and once in a while it also makes a mistake. So our program is able to filter out the actual, to differentiate the actual mutations from just the sequencing errors and artifacts. So here's just a broad overview of all the different programs I have written for this project. So this particular part detects the changes in DNA, and this little program over here filters out all the different artifacts. So now that we have all of these mutations in the DNA, we want to see which ones can potentially become a neoantigen. And if you remember, neoantigens are the antigens created by mutations in cancer cells. And these neoantigens can be bound by a T cell. So this prediction of which mutations can create neoantigens is complicated by the fact that there are thousands of possible MHC1s in humans. So this means that every one of us in this room probably has a different MHC1. And our MHC1s then bind to each antigen in a slightly different way. So even after we know which proteins have been mutated, it's still a hard problem to figure out which ones will actually bind your specific MHC1 molecule. So thankfully, this problem has been mostly solved for us by researchers who use this fancy-sounding artificial neural networks to do this type of predictions. So once we have a mutated sequence, we can say, okay, is this likely to become a neoantigen, or is it just a mistake in the DNA that doesn't matter? So when we look back at our patient who responded for the first 6 months and then relapsed, and we look at the neoantigen load, and here neoantigen load just means the number of genes that will become a neoantigen. We see that when the patient is responding, which is in this blue region, the neoantigen load is quite high. However, when the patient relapsed, the neoantigen load fell. And here the black dots means that this algorithm has predicted this peptide or this protein fragment to be a strong binder. And a gray dot means that the algorithm determined that this mutation will cause a fragment that will be a weak binder to MHC1. However, it's still predicted to bind. So something strange is definitely happening in this patient's tumor, and we decided to take a closer look. So the path where a normal cell becomes a cancer cell is not quite as straightforward as just normal cell gains many mutations and then it becomes a cancer cell. Because the cancer cell can still keep gaining even more mutations. So it might gain mutation and become this orange one over here, or it might gain some mutation and become this pink thing here. So we can label these different populations of cells, A, B, and C. And we can actually create what we can think of as a family tree. So in the very beginning, we have the normal cell. And then the normal cell gains many different mutations, and it becomes the population A. The population A then gives rise to B and C through separate mutations. So we constructed this type of family tree for our patient who had a relapse. And this is what the tree actually looks like. So we see that this patient had a normal cell that then branched into two very separate parts of the family tree. And one side we have the mostly green circles, and the other side we have the mostly red circles. So if we walk down this family tree, on the red side of the family tree, we find that very early on, a copy of the MHC1 has been deleted in this cancer cell. So what this means is that, so each one of us actually have two copies of MHC1, one from mom and one from dad. So everything after this point right here, every cell only has a single copy of MHC1 because one of them has been deleted. And then we found two separate mutations that completely destroy the MHC1 production. And these are two different mutations that occurred in the next two branches of this family tree. So this means that all the red populations on this tree have no MHC1 at all. So now that we have this family tree, we want to look at how frequently we actually see each of these populations in the tumor, and if this changes throughout time. So a tumor is actually not just a glob of cancer cells. It's a very complex mixture of these different types of cancer cells, but also some normal cells and some of our stormtrooper immune cells. So using our computer programs, we can delete the normal cells and the DNA from the immune cells out of this equation, and we can just look at our different cancer cells. And here the different cancer cells are represented by different colors. And we can look at how the fraction of these different cancer cells change in the tumor throughout time. So for example, we might start off with mostly these two yellow and orange lineages, but over time we see that we get more and more of this pink lineage. So what do we see in the actual data? Well, we see that when the patient was responding, most of the tumor consists of these green cells. So these are the cells that are on the left side of this family tree, and these have MHC1 expression still. So these cells are still making functional MHC1. However, when the patient relapsed, we see that these bars are now entirely red. And this means that these tumors now have no MHC1. So what does this mean in cartoon terms? Well, initially when the patient was responding, the T cells are recognizing the neoantigens on the cancer cells and killing the cancer cells off. However, during the relapse, now the cancer cell, even though it still has these neoantigens, it's not making any MHC1. So it doesn't have any of these hands on the cell surface anymore to show the T cell what it's making. So when the T cell comes by to check for identification, the cancer cell just says, there's nothing to see here, move along. Researchers are also looking at the predictors for who will respond to immunotherapy and who will not respond to immunotherapy. And currently, the research suggests that the more neoantigens you have in your cancer cells, the better your chances are for being a responder to immunotherapy. And this can make sense if we think about how the more neoantigens you have, the more likely you are to have a T cell with the right T cell receptor that will recognize that cancer cell. If you only have a few neoantigens, it might be the wrong shape. So when your T cell comes by, its T cell receptor doesn't fit that neoantigen. Researchers are also currently looking for other potential therapeutic targets. So the cancer cell can play this Jedi mind trick on the T cell in many different ways by using this blue molecule, or it can also use this red protein or some green protein. So researchers are studying different ways to block these interactions that might inhibit the immune system. And they're also trying to use these different therapies in combination because this seems to benefit patient survival. So in this last part of this lecture, we saw that T cells can recognize neoantigens on these cancer cells and kill them the same way that the T cell kills virally infected cells. We saw that our cancer cells can play the Jedi mind trick on the T cells by telling it that it's recognizing the wrong antigen, even though the T cell is recognizing the right antigen. Immune checkpoint inhibitors prevent the cancer cell from using the PD-1 and PD-L1 pathway to shut the T cell off. And finally, in our research, we found that cancer cells can respond to immunotherapy by just completely turning off the MHC1 pathway. And now, even if it does have neoantigens, it's not being shown to the T cells anymore. So ultimately, as we continue to improve our understanding of both immunology and cancer biology, scientists will continue to find more innovative cancer treatments that will both prolong life and also have fewer side effects. So with that, I would like to thank Science in the News and also to acknowledge all of these following organizations for their general support of our program. And I'll also take any questions that you might have. Yes?
How do they test for how many neoantigens a cancer cell has?
Right, so we typically do this by... Oh, okay. So the question was, how do we actually detect how many neoantigens a tumor might have? And we do this by taking the DNA from the tumor and also a normal blood sample. So we can compare the DNA from the normal blood to figure out where the mutations occurred in the tumor. And once we have these mutations, then we basically feed it into these computer programs. And we also know the patient's MHC1 type from the normal blood. So this program will then take the MHC1 type and also all the different mutations in the patient. And the program uses the artificial neural network to then figure out which ones are likely to bind to the MHC1. So it's really not predicting whether or not the patient has the T cell to recognize a neoantigen. It's just predicting whether or not this new mutated protein will be bound to the MHC1, because that means it will be on the cell surface. And once it's on the cell surface, then potentially the patient will have a T cell that's able to recognize it. Yes?
I'm just wondering how available is this kind of a process to your average cancer patient?
Right.
Is it only available to people who are in studies, or is it a more widespread?
I think right now it's still in more of a trial phase. I think it's mostly used for people who have really advanced metastatic melanoma, and it's also been approved for lung cancer and possibly a few other cancers. So I think it's pretty available through trials, but I don't think it's very commonly used right now. I think people still typically go through immunotherapy and some of the, chemotherapy and some of the other therapies available before moving on to this type. Yes?
I think her question kind of answered part of mine as well. But so if you're giving these T cells this checkpoint blocker, do patients end up with autoimmune symptoms then?
Yes.
Once they've sort of treated the cancer because the T cells can't?
Yes. Okay, so I just remember I forgot to repeat the question. So the question was that if we are treating the patient with these things that block these interactions and inhibit the immune system, does the patient then get really bad autoimmune symptoms? And the answer is yes. So I talked about how there was this 2011 generation of immunotherapy and the 2014 generation of immunotherapy. And the 2011 therapy gives you really, really bad side effects that is related to autoimmunity. And the 2014 ones actually don't give you really bad side effects. And this, the reason why this happens goes a little bit deeper into the biology. So the 2014 one, it targets a slightly different pathway than the PD-1 and PD-L1 pathway that we talked about in this talk. That pathway basically turns on all the immune cells in your body. So every single T cell ever, even if they might have slipped past that purple cell that was getting rid of the ones that recognize self-antigens, and they've been turned off in some other ways, now they're active. So they're all just kind of attacking whatever they recognize. The 2014 generation, PD-1 is actually only expressed on T cells that have recognized something. So the T cell goes and it says, you have a neoantigen, I recognize you, and then it turns on. And when it turns on, it then starts expressing PD-1. And the PD-1 is basically saying, turn me off if I'm wrong. So the cancer cell then says, you're wrong, turn off. So that's why PD-1 and PD-L1 blockers have fewer side effects, because it doesn't turn on immune cells that haven't been turned on once before already. So if that, okay. Steph?
I'm wondering, so what options are available in the clinic then for people that relapse on immunotherapies? Are there any other thoughts of like trying targeted therapies on top of immunotherapies, or what's going on with that?
Right. Okay. So the question was on what options are available to patients who have relapsed or didn't respond to immunotherapy, and it's tough. So usually these patients have already gone through several rounds of targeted therapies or chemotherapy, and it didn't work. So what I've seen in the clinic is that in some of the patients in our study, they go from one immunotherapy to another or a combination of the two. And sometimes people who don't respond to the 2011 one actually respond to the 2014 one, where they can give them a combination to see what works. And you could always go back to targeted therapy, but the choices aren't that available. And one of the targets is actually BRAF, which was the mustache guy that we talked about earlier. Yes?
I know that melanoma was one of the cancers that responded to BCG, which is the anti-TB vaccine. Not used in the United States much. But you know the mechanism? The immune mechanism for that? It's called the immune mechanism.
I'm actually not sure. So that's not really my area of research. So yeah, I'm not really sure about the answer to that one. Sorry. Oh, sorry. So what was the vaccine called? It was
BCG.
BCG. Ah, okay. So it was whether or not.
It worked by an immune mechanism of some sort.
Yeah, I'm sure. Okay, so it was that melanoma apparently responds well to the BCG vaccine, and he was wondering if I knew the mechanism for that, and I don't really, but I suspect it's probably something immune related, like you said. Yes.
You mentioned that if cancer cells have more neoantigens, there's a higher chance of them being detected by T cells. But if the MHC1 pathway itself is removed, then whether they have more antigens, the T cells will still not be able to recognize it, right? If it's not expressed on the surface. How,
So is your question why that the neoantigen levels fell during treatment for that patient? Or
if you have higher neoantigens, the T cells will be able to detect it. But if there's no MHC1, then won't it make no difference how many neoantigens they have?
Yeah, okay, so the question was, so if a cell doesn't have MHC1 expression anymore, then no matter how many neoantigens it's actually making, it's not going to go onto the cell surface, so the T cells can't detect it anyway, right? And we do think this is the case, so the loss of MHC1 is what we think is responsible for that patient relapsing. And I think this is also why neoantigens load by itself isn't a perfect predictor for who will and who will not respond. Because so the process itself is inherently very random, because the T cell population in your body is random. So even if you have a bunch of neoantigens, you might get really unlucky and you don't have the right T cell, or you might have a few and the T cell might recognize it. But then there's another layer of complexity where even if you do have neoantigens, you might not have the right, you might not have any MHC1, and then it's the same as not having any neoantigens at all. Yes?
Do you have a sense in your research with the MHC1 whether or not that population of cells existed already, and then you kill all the ones that had MHC1 and then that's why they become the larger population, or whether or not the therapy, or whether it's a new mutation that's acquired by cells that are already there?
Right. I think the general consensus in research is that all the cells were there in the beginning, and they're kind of existing in different populations. So initially, maybe 99.99% were the green populations, and then when you give the person immunotherapy, it kills the green population, and now because of all the green population is dead, the red population without any MHC1 is then expanding and taking over. So that's what researchers currently think, and it does seem to be, it's also supported by what we're currently finding in this research. Yes?
If there were a therapy that just killed all the cells in the body that weren't expressing MHC1, is that something that really only you see a lot of in cancers, or are there going to be a whole lot of normal cells in the body that just happen not to be expressing MHC1?
I see. Okay, so the question was whether or not you have normal cells in the body that just normally don't express any MHC1, and whether or not we could design a therapy that target these cells that don't have any MHC1 expression. So the answer to the first part of that question is no. Every single cell in your body should be expressing MHC1 at all times. And very early on in cancer cells, in cancer cell development, there's actually specialized types of immune cells that do target cells that don't have MHC1 expression, and it kills off those cells. And the problem is, like, during more advanced cancers, a lot of your immune system is kind of dysregulated. It's not functioning normally anymore, and that includes the cells that are targeting MHC1. So,
Joy, actually, small clarification.
Yeah.
Red blood cells do not express MHC1.
Good point, yes.
And you want those, so.
That's true. Okay, so excluding your red blood cells and the normal blood, do platelets express MHC1? I don't think. So like things in your blood, excluding the things in your blood, the things most of the cells in your body do. And also regarding current therapies, I'm not aware of any that specifically targets lack of MHC1 expression. It's inherently harder to target like the lack of something than to target something, right? So like if you have a BRAF inhibitor, it targets the BRAF that's broken. But when you don't have something, it's hard to really design something to detect that. Yes?
You had a slide up that looked like it was showing a flow chart of the workflow of information in your computational biology research.
Yes.
The programs created.
Is it this one?
That was, yes, that was to do your, this is you doing your experiment on that patient? Is that what this software is doing?
Okay, so the question was on what the software is actually doing. And it's not really the experiment on the patient. It's we get the patient's DNA, so now everything is just in the computer. So DNA just is like very long string of different characters. And these programs are basically aligning the different characters, figuring out where the mutations are, and predicting the neoantigens. So basically, all the slides that you see after this one, where we look at the family tree and how the different populations are changing over time, that's the result of all these programs here.
And what work were you doing on this project? It sounded like you said something about writing some of these programs as part of the project, and I'm just trying to understand what goes in and out of them, and did you build parts of them? How did you build them? How do they work?
I see. So the question was about my what my actual role was on this project and whether or not I wrote some of these programs. So, right. So this is an interesting project because we have someone who was in charge of all the experiments. So he was at the hospital actually physically getting these samples, breaking up the cells and getting the DNA, and then getting the DNA sequenced. And then I'm the person who does the downstream work. So the DNA comes to me, and then I run it through these programs, and I also wrote a few of these. So these programs kind of exist as separate entities, and I went in and connected them together. So it's kind of like the input is a DNA, and it goes into one program, and then the output from that program then goes into another one. So that's why these things are kind of connected by these little arrows. And yeah, so I guess to answer your question, I did the computational part. I did not write all of these programs, but I wrote some of them. All right, well, thank you very much for coming to the lecture.